Hello friends, today I am going to discuss with you one of the most important pathway of complement protein activation, that is classical pathway. We know that this complement protein is generally present in the blood or in serum in inactive form, and they get only activated after antigenic challenge or entry of antigen. Now there are three different pathways by which this protein can activate, like classical, alternate or lectin pathway. In today's lecture I am going to discuss about classical pathway. Now, the complement protein in the human body can be named as C1, C2, C3 up to C9. They are generally present in zymogenic form or in inactive form, and after antigenic challenge, they get activated by stepwise manner. Finally, due to activation, they eliminate the pathogen or microorganism from the body. Now, classical pathway initiate when any antigen enters in the body which combine with the antibody. Mainly IgG1, G2, G3 or IgM antibody play vital role in the activation of complement protein by classical pathway. Now we know that antibody can bind to the antigen on a special point or special site called epitope, and antibody having two different parts, AB that is fragment antigen binding and FC that is fragment crystallizable. After binding of antibody to the antigen, the first complement protein called C1 protein bind to the FC portion of the antibody. The FC portion of the antibody having a special type of a receptor or complement protein, but only the receptor get proper confirmation when antibody bind to the antigen. Once antibody bind to the antigen, C1 protein is ready to bind to the receptor which is present on FC portion of the antibody. Now this C1 protein having three type of subunit, C1q, R and C1s, out of which Q subunit of C1 protein bind to the FC portion of antibody. After binding, the C1q protein get activated and then it activates C1R protein by hydrolytic cleavage. C1R protein is nothing but the serine protease enzyme which finally activate C1s protein, which also function as serine protease enzyme. The C1s protein having two substrate, mainly C4 protein and C2 protein. C1s protein cleave C4 complement protein into two parts, C4b and C4a. Out of these two parts, C4a protein diffuse in the blood and deactivated, while C4b protein go and bind on the surface of an antigen. Now this C1s protein again cleave C2 protein into C2b and C2a, out of which C2b protein diffuse in the blood and get deactivated, while C2a protein bind close to the C4b on antigen. Now a complex is produced on antigen called C4b C2a complex, and this complex which is present on the antigen surface is called C3 convertase complex. We called it as C3 convertase because this C4b C2a complex having ability to cleave C3 protein into two parts, C3b and C3a, out of which this C3a part of C3 protein diffuse in the blood and get deactivated, while C3b part of C3 protein go and bind to the C4b C2a on antigen surface to form a complex called C4b C2a C3b complex. This complex is called C5 convertase complex. This complex having ability to hydrolytically cleave C5 protein. So this complex, that is C5 convertase, cleave C5 protein into two parts, C5a and C5b, out of which C5a part diffuse in the blood and get deactivated, while C5b is one of the most important protein of this pathway which activate remaining complement protein, that is C6, C7, C8 and C9. All these protein go and bind to the antigen surface on some other site and form a special type of a complex called MAC complex. MAC is a membrane attack complex. This complex on antigen surface form pore or hole or ion channel on antigen surface. Through this pore, important ions and minerals are loses or come out from the antigen surface, and then the osmotic balance of the cell, of the microbial cell, get disturbed, which is going to finally lead death of antigen. In this way, the classical pathway is operated which finally end with death of the antigen. Thank you.